Well, good afternoon and welcome to the third speaker in our first, hopefully, annual TLO Golden Speaker Series. And really glad to have you here, both you, those of you who are here in person and remotely. Our third speaker is, actually it's a husband and wife team, Professor Kristala Jones Prather, and her husband Darcy, who have been involved in an entrepreneurial spin-out of work from Chris's lab at MIT. And we'll be speaking from her first, and I prepared an introduction for her, and then I just got Darcy's bio a moment ago, so I'll be reading that off my phone. But first of all, my name is Ben Rockney. I've been with the TLO, the Technology Licensing Office, for about 8 years. My portfolio primarily consists of medical devices, and the PIs that I represent and help lead their IP through patenting and licensing are primarily from the departments of from the Institute of Medical and Engineering Sciences, or IMES, the Media Lab, and the McGovern Center. In contrast, Chris is from none of those. She's from the Department of Chemical Engineering, and I very much enjoyed working with her for my time at the TLO. And she's actually been quite a prolific inventor. She's disclosed 38 discrete inventions to the TLO, of which 42% are licensed, which is, from the TLO's point of view, the real success story. So we're really glad to have you here with us. She's the Arthur D. Little Professor in the Department of Chemical Engineering and is also the executive officer of that department. She received a bachelor's degree from MIT and a PhD from UC Berkeley. And after her PhD, she worked for 4 years in the Merck Research Laboratories and then joined around 2003. She joined the MIT faculty. Her research interest, which she'll be at MIT, which she'll be talking about today, her interest is centered on the design and assembly of recombinant microorganisms for the production of small molecules and plasmids. And she also focuses a lot of attention on designing novel bioprocesses and biomanufacturing. She's a recipient of numerous national awards from the National Science Foundation, the American Institute of Chemical Engineers, the American Association for the Advancement of Science, and the American Institute for Medical and Biological Engineering. And as I said, she's a co-founder of a spin-out from her laboratory named Calion Incorporated. It was an MIT licensee built on technology developed in her lab. She's the scientific advisor for 4 other small to mid-sized companies, as well as a Fortune 500 company. She's on the board of directors of Biomedical, a manufacturing innovation institute focused on advancing domestic biomanufacturing. And very recently, really probably 3 or 4 months ago, the TLO licensed one of her bacterial strains to the largest domestic, I think the largest actually worldwide provider of DNA plasmids, who'll be using the strain that she developed in her lab to achieve higher yields on their main production line. So we're excited about that license. Joining her this morning then, as I said, is her husband, Darcy Prather, the CEO of Calion. And at this point, I'm going to flip over to my phone. And like Professor Prather, the company Calion focuses on creating renewable chemicals in a more efficient way to make the climate safer. The EPA has awarded Calion and its team their prestigious Green Chemistry Challenge Award for their work with glucuric acid. I think she'll be speaking about this. Glucuric acid may change how we manufacture and recycle our clothes, among the many other uses that technology enables. Through his work with Calion and as a board member of the Green Chemistry Commerce Council, Calion hopes to ensure a safe and vibrant environment for generations to come. Mr. Prather earned degrees in electrical engineering as well as science, technology, and society at MIT. As an undergraduate, he was a member of the football team, and I didn't know MIT had a football team. They play Wellesley, right? Anyway, he was a member of the football team, very active in the National Society of Black Engineers. He studied philosophy, politics, and economics at Oxford University after winning a Rhodes Scholarship. Subsequently, he stayed engaged with MIT with various stints as a member of the MIT Corporation, their board of trustees, and a member of the MIT Alumni Association board. He started his career as a management consultant at McKinsey, spending significant time studying R&D and he's worked in various small companies until he co-founded Calion. And he and Chris have, it says right here, 2 amazing daughters. I know one of them just came down with COVID, though Darcy and Chris have tested negative, but that's why they'll be doing their talk with masks. But we hope a quick and speedy recovery for their daughter and safety for them. So with that, please please join me in welcoming first to the stage, Chris. Thank you very much.
So let me start by thanking Ben for the introduction, and thank you for being here, and to those of you who are online as well. We're going to hold questions until the end of the presentation, and there'll be plenty of time to ask questions both in the room and for the live stream audience. So we're going to do this as a tag team presentation, and I'm going to immediately ignore one of the guidelines that Ben and Robin gave me, which is to tell stories and don't talk about technology, but I'm an MIT professor, so I can't do that. So I'm gonna talk about the technology more at a higher level. There's not going to be an exam afterwards, and there won't be too many details there. But then I'm going to switch it over to Darcy to really talk about the company. And so I really wanted to be able to give the story of how this started in a lab, and then how it transitioned, and the interactions and engagements we've had with TLO over that time. So can you guys switch me to the slides here in the room? All right, so the first thing I want to point out is this note in the lower left-hand corner of the slides. It says that the presenter declares competing financial interests. That is something I have on every external presentation that I give. Those competing interests are because I'm a co-founder of Calion, as Ben has mentioned. And I'm happy to take questions about this at the end. I'm not going to spend a lot of time here, but as an MIT faculty member who also is a co-founder of a company, I do have to have a conflict of interest management plan, which is on file with both the VPR office for Vice President for Research, as well as the department head for chemical engineering. And so if anyone's curious about the implications of that, I can talk about that later, but I was asked to very explicitly make sure everyone knew I do have a COI management plan. So let me start with the overall objective of what we do. And the context for this is in using biology to do chemistry. And so, again, I said these are not going to be very technical slides. This is just a reminder of the fact that we as humans have relied upon the ability of biology to do chemistry for really thousands of years, for all of civilized humanity. This is an example that perhaps some of you are familiar with, which are beverages, in which you actually have alcohol, in this case ethanol, being produced from sugar. And that is actually mediated by a microbe, by yeast in this particular case. And so you have sugar being converted to ethanol. Those are words, but those are actually also structures. Glucose is the primary sugar, which is then converted into this 2-carbon molecule ethanol. And as a result of this reaction, you have to have some chemical equation. Everybody remember chemistry in high school? People get nice, warm, fuzzy feelings. I had to take a note. But you essentially recognize the fact that there's some relationship between the inputs, in this case glucose, and the outputs, which is ethanol. And that happens as a result of DNA, where DNA is encoding RNA that's then translated into protein. And it's those proteins, in particular proteins, enzymes, that allow us to now perform these reactions. And you actually have not just one enzyme, but many, many, many different enzymes that are doing these reactions, okay? It also turns out that that happens inside of a cell, and inside of that cell is actually not just one reaction, which I gave as a overall summary reaction of glucose going into ethanol, but really there are a lot of different chemical reactions that take place in cells. And the take-home message here is that as scientists and engineers over decades now, not quite thousands of years, but over many decades, we've been able to exploit this to bring to market molecules that are useful and of interest. And so some examples here are in the medical space. What I have in the upper left here is the structure of something called lovastatin. It was the first FDA-approved cholesterol-lowering drug, which is the result of a fungal chemical reaction. There's an organism that makes this compound. There used to be many, many more efforts in big pharma looking at so-called natural products, which are chemical structures made by a variety of organisms. and testing them for their ability to be used as therapeutics. So that's one example. On the upper right-hand side is an antifungal compound, something that was one of the more recent, actually, antimicrobials that have been produced. The canonical example of this is penicillin, which was discovered in the 1930s and was really manufactured at large scale in the 1940s and was considered to be a game changer for the Allied forces during World War II because injured soldiers were very likely to have bacterial infections, and those who were treated with penicillin had a much, much higher success rate of recovery. So the point here is that we have been able to take advantage as a society of this ability of biology to do chemistry. We've also been able to take advantage of our ability as engineers to scale these processes in order to get really, really large quantities of things that we need, and importantly, to be able to produce them from renewable sources. Okay, I have on the bottom of the slide 2 other examples, and there's this is a slide which is much longer technical talk, but I had said I wasn't going to completely ignore Ben and Robin's advice. But the main point here is that you can use biology to make things that are very, very complicated, like the 2 molecules at the top, or things that are a lot less complicated, but also still very important, and much more economically produced, or produced much more economically through fermentation or through biology than through chemistry. Okay, so that's sort of background. What I want to draw your attention to now is what we do in our lab with this idea and where the motivation for it came from. And it really comes from climate and from the fact that if we think about where we get most of our stuff, I'm a chemical engineer, and when I try to explain chemical engineering to our first year students, I tell them chemical engineering is about stuff. It's about designing stuff, it's about making stuff, it's about really being able to figure out how do we get access to the things that we need. And the reality is that a lot of the stuff that we need, and some stuff may be considered frivolous, but some stuff is really, really critically important. A large fraction of that comes from non-renewable sources. So I have an example here of crude oil being used as an input, and we're accustomed to thinking about oil as an input to give us fuels. And that, there's a pretty clear connection between that. But we're less cognizant of the fact that the carpet fibers that I'm standing on now and that your feet are resting on come from oil. The plastic that covers this clicker comes from oil. The casing on my laptop comes from oil. And so if we think about now that part of it, it's a slightly different problem, because if I'm only interested in energy, I can say, well, we can get rid of gasoline by converting all of our cars to being EVs, right? We're not going to talk about where the electricity comes from, but I could say, I can get rid of it that way, right? I can say we're going to get rid of our gas stoves. You probably heard that in the news recently, right? Right? And we're going to go to electric stoves. But that's replacing now the energy component of crude oil with alternative sources of energy. You can only replace mass with mass. And so if we're going to say we're not going to rely upon oil to get the carpet fibers or to get the plastic casings, and even if you can think about many other ways in which plastics are made, or you can replace plastic with paper, but there are lots of industries, for example, medical devices, which Ben mentioned, which are critically reliant upon the unique properties of plastics. So this presents a different kind of problem, which is now we need alternative sources of mass to be able to give us the materials that we need for life and for quality of life. So we ask ourselves then, where can that mass come from? And that's biomass. It's a renewable source where now CO2 is being taken out of the atmosphere in order to build plant matter. And we can think about deconstructing that plant matter and having it be a source now to make, for example, biofuels. But we've been much more interested in making other chemicals, okay? And that's because if we think about the price points you have to reach for biofuels, if you think about the scales that you have to reach for biofuels, it just never really made a lot of sense to me to do that first, right? It makes more sense to figure out how you actually get the technology to scale appropriately with something where you can make less of it and you can make it and sell it at a higher price than going for biofuels. And so when I started my lab, and coming from Merck, the whole point was to figure out, can we actually use biology more to do more chemistry and different kinds of chemistry than what had been done before. So this is just one quick slide to sort of give a sense of how we think about this. So if there's a compound that we know we want to make, for example, this is showing I'm starting with A and I'm going to go to D. Then I have access to lots of different tools that say, well, let me get rid of those parts of the pathway that are not productive. So this is showing this side stream here going to K. And then let me overexpress or re-engineer enzymes in order to have the pathway be faster overall and more productive. I can also think about taking advantage of the fact that with biology, if I go back to that first example of making ethanol, there are a series of enzymes that are important for making that, and those enzymes are encoded by the DNA, and I can get DNA from anywhere. So if I know that there's DNA in some weird organism that got scraped off the bottom of my shoe, if I know what enzyme that encodes and what chemistry that performs, I can then mix and match those from different places and now make an entirely new pathway. And then another way that we think about this is in actually designing the chemistry itself. And this relies more upon traditional ways of thinking about chemistry as just series of chemical reactions. And we think about making new kinds of products based on existing types of reactions operating on new kinds of substrates. Doesn't matter if you didn't understand anything that I said there. I'm going to summarize it and say there are a few different examples we've had. And what I have on this slide is just giving you a sense of the complexity where I have number of genes and number of organisms. Genes now are going to be the number of enzymes. Organisms are the number of those sources of biology that we're using to take this material from. So hopefully you can see from this that we have a way of thinking about making different kinds of molecules and making them in different kinds of ways, okay? That's as complicated as the science is going to get. And on the right-hand side, of course, I'm writing down or summarizing some of the motivations I've given you before. So just very briefly, I want to start walking you through our process of taking that as a concept and then moving that into Callion. Glucaric acid is one of the compounds we've worked on. Ben mentioned it in his introduction, and Darcy will talk about it more. I first identified glucaric acid as something to work on because there was a report from the Department of Energy that labeled as a top value-added chemical from biomass. And I'm going to come back to that in a second. It turns out it is a natural product. It's made in fruits and vegetables, and it has been found in human urine. So it's a product of human metabolism as well. And when we started our work on this, there was no known microbial pathway to be able to make this. So if you go back to that image I had with all those dots and all those different lines, if I know that it's found in fruits and vegetables, there is a series of those dots that allow me to get to the product, but it's much more complicated than what we could think about putting together in a scalable way. It is used in the synthesis of pharmaceuticals in making Adderall, actually generic Adderall. And there are many other uses that have been demonstrated as proof of concept, but are really limited by lack of access to sufficient quantities of this compound. There is a process to make it chemically. There were actually when we started our company, 2 other companies that were making it chemically. Neither of those exist. So by the way, you don't have to be first. Better to be last because you managed to stick around, right, than be first and not be able to get through it. And the problem with chemical synthesis is both that it utilizes nitric acid in very large amounts. So in a world where there's a lot more attention being paid to green chemistry and environmental impacts, having a process which produces this very, very highly corrosive and very toxic waste stream is a big disadvantage. And the purity that you get from using the chemical process is not so great. So some of the applications you may think about using it for, I mentioned materials before, carpet fibers, those are polymers. And if I want to use something in a polymer, it has to be actually pretty pristine. The kinds of specifications you need from monomers into polymers, in many cases, are more stringent than what you need for a drug. Right? Just think about that for a second, right? So the specifications for your carpet fibers might be more stringent than the specifications for your heart medicine. Okay, and that's just because of the chemistry that's involved there. So we were interested in making this compound, and I will be honest, we got motivation from the DOE report. It wasn't in my mind necessarily thinking about a company, but I thought, I'm going to try to get tenure at MIT. I got to do something that people say is hard. So let's look at some stuff that have been some targets that have been identified as good things to do, and where people don't really know what the answers are to that. So we started working on this in the lab. I came here in 2004. My first student started working on this project in 2005, and we had some fits and starts, and we got some stuff to work. And where TLO got involved was that I got invited to give a talk at a conference at the University of Illinois, and you can see the date on here is April 5th, 2008. And Ben mentioned that I had spent some time working at Merck, And fun fact, one of my tasks at Merck, we had a whole group that was looking at bioconversions or biocatalysis. So looking at new kinds of reactions where we could use biology instead of chemistry. And people on the team had different tasks or responsibilities. And I got tasked to the IP group. Right? So my job was to sit down with a lawyer at Merck and try to understand what freedom to operate, what the FTO was in this space. And it was a really fascinating experience for me. I learned something very important. I learned that a patent gives you the right to sue. Right? You think about a patent as giving you ownership, but what it does is give you a leg in court. And so I would ask him, is this a space where we can operate or not? And he would go, I don't know, right? There's a patent. Could the patent stand up to prosecution, to litigation if we took somebody to court? I don't know. So I learned that the space is fuzzier than I think we are instinctively taught to believe, or we instinctively fail to believe. But I also knew from that experience that there are some timelines associated with when you need to file relative to when you're going to make a public presentation. So it wasn't Ben that I reached out to, but there was one of Ben's predecessors at TLO. About a month before this, I emailed and said, I have this presentation I have to give. We've been working on this thing. I think it could actually be useful, right? I do think it's novel. And I have a slide in my slide deck where I'm going to demonstrate that it's been reduced to practice. And I know from my time at Merck that I need to have a provisional patent application filed before I give this talk. Now, we had actually started writing up the paper for this, and that turned out to be great because I had the cover sheet for submitting a disclosure to TLO. And then we just included with that the first draft of the manuscript. And so if you go back to what the patent specification is, a lot of that language you can find in a paper in Applied in Environmental Microbiology, where we first reported the synthesis of this particular compound. And so we worked with them for a few weeks, and then the first provisional patent application was filed. And you'll see the date on that is April 4th, 2008. Remember, the talk was on April 5th, 2008. It just has to be before. Actually, I think technically it can be the same day. Right? I don't know if it's by midnight or by 5 p.m., but it can be the same day. But this talk was on a Saturday. So the provisional patent application got filed on a Friday. Okay, and that's great. You can see my two co-inventors here. We're pretty happy to have a disclosure. But once you do that, the clock starts ticking. Okay, and so to be perfectly honest, we submitted this provisional patent application, and then we went on doing other things with the lab, sort of going on with life. And then about a year later, as the question arose about whether or not this was something that should be converted into a full patent application, you then have this decision to make. So a year later, again, this is the report that I referenced in terms of getting those molecules from. I get an email from TLO saying, is there any interest? Do you think there's interest in licensing this? I actually gave this report to my husband, who I handed off to in a couple of slides, and said, I got this email from TLO. What do you think? This report, by the way, the objective of it was to try to identify something of higher value than fuels that could be produced along with bioethanol or some other alternative forms of biofuels to be able to increase the overall value of a biorefinery. And they had come up with this list, and I mentioned glucaric acid, but there was actually a second compound on that list, hydroxybutyrolactone. It was on a slide, or the structure was on a slide a few slides ago, and we had actually shown that we can make both of these. And so at the time when we were having these conversations, there was both this question of what do we do about the glucaric acid patent, and then is there interest in bringing, as we would call 3-HBL, to market. So I'm going to do one more slide before I hand it off to Darcy, but it really was an interesting process to try to figure out, should we go forward with this? And I'm going to let Darcy tell that part of the story. The one thing I will say is that, actually I'll ask you, how many of you are students or postdocs? Okay, so, and you're all going to be rich because you're going to make companies. Remember how wonderful MIT is as a place. And when you make $1 billion, I'm looking at you. We'd love to name something after you. For right? I didn't have any students or postdocs in my group who were interested in going with the company. So this happened around the same time that my first students were graduated. I had two of them who were graduating. I had one who was actually one of the co-inventors on the glucaric acid patent, was very focused on an academic career. And so he had his postdoc lined up, and he was ready to go. And the other one who actually produced the 3-HBL work just was like, startup sounds way, way, way too risky to me. He went to work for Dow, which then became, actually he went to work for Roman Haas, which then became Dow, which then became Dow DuPont, and is now DuPont. So as he says that he's worked for 4 different companies without ever leaving his office, right? And so he wasn't interested in that either. And so, again, I'm going to have Darcy tell this part of the story, but it is an interesting difference, I would say, versus what typical MIT startups will experience, which is that usually you have an inventor on the work within the lab, or at least someone who's a close collaborator within the lab who's interested in that. But we did decide to actually go for it. And so this is actually the first patent that was issued, and you can see the priority date, or the PCT filing date here is April 3rd, 2009, so one day before April 4th, 2008, which was that 12-month window that we have there, okay? So I'm gonna hand it over to Darcy to give more of the story of how Calion came to be, and then again, I'll come back and we'll both take questions.
Hi, everyone. So to finish off that little bit of the story, Kristala, having worked at Merck, one of her colleagues was at a conference with her, and she gave her a talk about glucaric acid, and he goes, "Oh, are you guys starting a company?" She calls me back, reports that, and I was like, "So can he be CTO?" The answer is yes, and then ultimately that was the start on the technical side for Calion. So let's see here. Yeah. So I'm just going to give you guys, for some of you, just some perspective, right? How do you launch? How do you make this decision about starting a company? First off, you got to throw out, does anyone care about what you're doing? And maybe it's just you who cares about what you're doing, but maybe there's a billion people who have no idea that they should care, but you can know for them. But in the end, you got to throw out, who cares? You got to build a team we were just talking about. Couldn't have started without Neil Connors. Having thought about at least a couple of team members, you got to get lots and lots and lots and lots and lots and even more advice from folks. Because you're never going to be the first person who's encountered some issue that you have when you're starting a company. VMS is by far the best resource any university gives you access to in helping you understand how to start a company. They were terrific when we first started talking about this in the late noughts, and they've only gotten better. So it is absolutely a phenomenal resource, and one of the reasons why the risk profile of starting a company at MIT differs then from other places, because you can tap into the advice, and those hard learnings don't have to be as hard. But ultimately, you got to create something that you can actually show to an investor, to a company, to one of the billion other people who should care about what you're doing, and scale it. And keep in mind, you always got to be ready to change. You don't know why. It could be your team. It could be your products. You mentioned the 2 products there. So we had glucaric acid. We had 3-HBL. 3-HBL, it turned out one of our advisors was another professor whose former student ran the largest generic manufacturer of statins in India. And our whole premise is that you could use the chirality of the 3-HBL to lower the cost of producing a purely chiral unit that's needed for statins. He called his former student, and his former student goes, "Yep, we cracked that problem." Because it used to be you had to drop it to minus 40 degrees C. In order to do that, very energy intensive. They said, we cracked it. So we said, never mind, we'll move on, focus our energies on glucaric. So why glucaric? Well, A, it was in that report, right? So this report said, oh, it can be used in all these different places and it could be quite amazing. But of all the things I'm listing up here for you, the only one was actually the nylon 6,6. That was the only one that was known at that time. But as people started to play with it, they discovered other new and interesting properties. As we were able to provide people with high purity glucaric acid, then where they were able to run very different experiments than they could previously. So she referenced earlier the pharmaceutical grade versus a chemical grade or a textile grade. In the space of textiles, we were able to provide a professor at NC State who recently got tenure based on her work with glucaric acid, where she discovered you could add small amounts of glucaric acid to renewable textiles and other textiles. So things like rayon or viscose. Turns out rayon, unlike nylon, is actually from trees. So rayon's renewable. But renewable fibers that come from trees tend to be much weaker than your synthetic fibers. Well, with her work, we can increase tensile strength 50%, we can increase toughness an order of magnitude, we can increase modulus about 100%. There are these huge leaps in the properties of textiles. Traditionally, in any of these fibers, people would be excited about a 10% change. So being able to discover these sorts of things, because we're able to provide a new form of glucaric acid that was much higher purity, and ultimately for us, you're always looking for the simple way to connect with someone. We'd say, well, we're gonna produce this as cheaply as beer. You ferment beer and wine, now you can ferment glucaric acid. And that's sort of the simple pitch that we would talk to people about. So, if you've got something that you think people should care about, next you gotta build a terrific team. So starting with Kristala, one of the things that ultimately we ended up doing differently was that from day one, Neil Connor said, you know what? I think we don't need our own lab. I think we can do this all virtually. Now, we had a lot of problems and challenges actually along the way, convincing people, because the entire world of VCs is about pattern matching, right? So my company will be the same as X, but in Y. And that's what they're looking for generally. So for us to say, we're gonna give you this new product that no one uses, and we're gonna do it in a different business system way, that was challenging for folks. But we ultimately did it. It was fascinating. So Neil we got some experimental results. He looked at it and he said, oh, you've got a loose piece of tubing. The people in Georgia, he was in New Jersey, I was in Boston. He's in New Jersey, and they go, what? It's like, yeah, there's some hosing that's loose somewhere in your system. Sure enough, they go back in the lab, they're like, oh yeah, there's a loose piece of hosing. Boom, ran the experiment again, nice results after that. But we could only do that because he was very experienced, right? So he had 20 plus years at Merck by that point. Tyler Smith, who ultimately joined us later as a team member now, actually came to us from one of the other glucaric companies that were not as successful. We talked about these purity issues. Their process, which was a nitric acid oxidation, would only yield around 30 to 40% glucaric acid on a dry weight basis, versus ours being 95 when we don't do things well, and when we do things really well, it's 99 plus. So things like that. And then other advisors, we had an SAB board, but there are lots of people beyond the SAB board that we talked to. Charlie here at MIT, sort of a legend within the chemical engineering space. He was one of the founders of Genzyme. So for us, we get a lot of lessons of him from those early days in biotechnology. And now we're saying, what does it look like for industrial biotechnology? And how do we think about those things differently? A simple example there is, many of you might be like, I can sit in front of a computer and I can design a new compound, and I'm gonna give you a platform that can design lots of new compounds. That was the same thing that people thought about when we first discovered how to manipulate DNA. They said, oh, it's a platform. People are gonna come running to me and they're gonna ask me to design things, and then we're gonna give them the design, we'll just get a license. Turns out big companies don't believe small companies can get things done. And so those models that fundamentally rely upon you being a platform in that way actually tend not to work. On paper, they look terrific. There's a company in our space called Biomedical that had that as their early premise. And ultimately, in order to prove that your platform really works, you've got to build your own product. That's what Genzyme actually did. They thought they were going to be a platform. They decided to become an actual product company. And it turns out if you actually look at the largest companies in the world, typically 1 or 2 products deliver 50% or more of their revenue or profits. And that's true in pharma. If you look in the chemical industry, there are what we call platform chemicals. And all of their chemicals are going to be derivatives of these 1 or 2 platform chemicals. And that's kind of how the chemical companies divide the world up. Whether they have access to polyethylene or polypropylene, they just divide the world up in that way. But we have all these terrific advisors, and that was super important because, again, they shortcut things for you. They also allow you to tap into other resources. They make people aware of what you're doing, whether it's VCs or other expertise. Because you're going to have expertise in whatever you've been trained in, and there are just so many pieces of expertise that you need to build a company. So being able to access trustworthy, reputable people is incredibly important. So for us, ultimately, we ended up going down the water treatment opportunity. It turns out one of our competitors, who's no longer is in business, they actually flushed this opportunity out for us, and what we thought was phosphate replacement initially. So phosphates are harmful to the environment, but they're commonly in the effluents from factories because they're used as corrosion inhibition. Turns out in hard water, the calcium that's in that water will coat, let's say, a reactor. Well, a reactor is suddenly coated with another chemical, you lower its efficiency. The energy cost of that are much more important than the cost of what's typically used for your water treatment. It turns out you get tremendous process economics by doing it. So our first customer ends up being Baker Hughes, who manages oil refineries for various folks around the country. And what you're ultimately looking for are customers, of course, who can say nice things about you. So we've been able to get folks in water to start saying nice things about Calian, and that's what we're driving towards at the moment. So you'll spend lots of time trying to get yourself ready to launch. You'll spend a lot of effort, again, trying to find those customers and those uses for things that you do initially, and most of your first year, people will pound you on, do you have something where people, once they realize it exists, they can't live without, right? Often the question is, do they realize that they miss it, right? So when people talk about smartphones, right? No one thought of smartphones, but once you were told what it could do, then you're like, oh, I gotta have that. So there's often a lot of education that's required. And then ultimately, you've gotta produce at scale. So for us, that was, again, one of the innovations that we had in terms of our approach. Within our space, typically people would spend around the order of $50 million to build a pilot plant, which would allow them to produce ton quantities of materials. It would not be economical at scale. It still wouldn't be at an economical scale, but it would be enough so that their customers could actually test and validate the product. Within our context, I'll show some pictures later, we were able to do some toll manufacturing. So we got both it at scale without spending $50 million, or the scaled production facility, which would cost us around $300 million in order to produce our first products. And again, but having that scale allows you to do a lot of activities with your customers. So for Baker Hughes, they bought our first few tons of product. They're testing it now. They're revalidating that it actually works as they expect and as we expect. And then they're like, okay, we're off to the races once that happens. We talked a little bit about other people. One of the fascinating things you'll find is VCs will ask you, okay, who are your competitors? Are you unique? Now that's the question I always find fascinating, because the world has existed without your product, which means whether people have it or not, there's something else that they could be buying rather than your product. There's always something that can be substituted for your product. And on the flip side, if you're doing something interesting, if you're the only one doing it, then there's some question as to whether it's actually as interesting as you think. Because ideas spread, right? That's the nature of universities is, hey, look at me. There's this amazing thing that I now know about. So with glucaric acid, when that report came out, several different companies got into the space. Ourselves, Rivertop, Renovia, for us, one of the interesting things would be in the conversations with potential investors, they would say, well, is yours going to be the lowest cost? Now, some people say to our benefit, sometimes it's to our detriment. We're very direct. And like 15 years ago, we were basically like, there is no way anyone could possibly tell you whether our process is going to be the cheapest, whether Rivertop's will be the cheapest, or whether Renovia will be the cheapest. Rivertop would tell people, oh, nitric acid, super cheap. We just throw it on sugar, super cheap. We are the cheapest process. And we go, fine if you believe them, but A, they can't produce the chemical, and you haven't even started to talk about the DSP, or the purification train, and how expensive is that gonna be? And you really don't know until you start scaling how cheap or expensive things are gonna be. Turns out these other methods, those purity issues ended up killing them, and they still couldn't produce at the cost that they were talking about. So ultimately, they died. However, they left a legacy of new potential applications. So for us, we're looking at these additive applications. For Rivertop, they were looking at the water treatment applications. For Renovia, they were looking at being able to create renewable nylon 6,6. Nylon 6,6 is the largest class of plastics, essentially, that we use in the everyday world. So you could go from glucaric acid, they showed you could go from glucaric to adipic, and adipic is the principal precursor to nylon 6,6. So there's still this legacy that they've left from which the whole world benefits, whether Calian ultimately ends up being the lowest cost producer. There's a new company that came around called Solugen, also related to MIT. So we've got to compete against them because it's still a great idea. So there's always going to be the next competitor. But against those competitors, we said, we've got high purity, we've got high yield, we've got this short, simple pathway, which is important in the biospace, et cetera. So we're able to produce a variety of different types of salts from glucaric acid that ends up being very important if you're trying to do different types of applications. This is a partner with Evonik. They're in Slovakia. So we literally looked at hundreds of facilities in the US, Europe, and essentially the world before landing upon this partner. Evonik is one of the largest chemical companies in the world that you probably have never heard of. They do on the order of $20 to $25 billion in annual revenue. There, we successfully scaled up. This is the calcium salt. This is a counter ion that goes into Adderall. Turns out the active ingredient in Adderall is just amphetamine with some counter ions to it. This is the water treatment product. So all of those totes are products from our last run that we did this past summer in August. As the first time we went out there, we were worried about COVID. The second time we went out there, we were worried about a global war erupting, but all went well. And then just to loop it back to the very beginning, Kristala was talking about, oh, you can go from sugar to ethanol. So lots of people do that. We've spoken to them about, well, if you're processing bushels of corn, what's your margin on that bushel of corn? They told us it's $5 to $8 a kilo type of margin. Like, well, what if you valorize that or create additional value? Well, with glucaric acid, you get about 10x that for each bushel of corn that you end up processing. So we got them to at least start to thinking about why they should care about renewable chemicals. So I think that's all I have for you. Here's a little timeline of starting at Kristala's work in the strain development back in 2005, 2004. And then we had to do some lab scale fermentation. So she published that paper. We're probably 10 to 30x, depending on how you think about it, in terms of what she produced in the lab. Then we had to do some downstream processing. It was all chemical engineering, but it was completely unrelated in some sense to what she did at the lab. And then we had to scale that up. So 1 liter scale for that lab scale. Pilot scale was about 300 liter tanks, 300 to 500. And then commercial production for us was in a 50,000 liter tank. And if all goes well, we'll do another 5 to 10x that a few years down the road. Also along the way, we won what used to be the presidential green chemistry challenge. We had a president who didn't care about green chemistry, so we removed the presidential green chemistry challenge. And it's a certified bio-based product. So bio-based products, of course, is becoming more important, but it's a recognition of the important work that we have done. So I think that's it for me. Kristala's going to come up, and I guess we'll answer questions.
Question. Can you talk a little bit about the fundraising track that you guys had to endure, I'm sure. I think I saw your name on Launchpad, if I'm not mistaken.
That's right, so Launchpad.
So that's what the name kind of rang a bell. So
See, that's why he's here, because I wouldn't have any idea how to answer that question. You're up.
So fundraising. Yes. The question is, can we talk a little bit about the fundraising that we had to go through in order to get to this point? So fundraising started with VMS, essentially. So by connecting with VMS, they helped us clarify the way that we should do our pitch deck. Almost all technical people, by nature, and I count myself in this, want to tell you how great the technology is. Almost all funders only care about why should they care? Why can I sell this very profitably in an exclusive way? So they helped us ramp up. They also gave us a connection to an angel group called Cherrystone, who had had a recent, very successful exit out of MIT's chemical engineering department called Smart Cells. Smart Cells. They were based in Rhode Island, but one of their members was part of VMS. He took us down there. We presented. They're like, oh, interesting. Well, how far is Kristala going to do some more work? So they made us come back like a year later. We presented again. That time they decided to put in some dollars, and that sort of initiated us being able to reach out to other angel groups in the area, including Launchpad, including Golden Seeds. Golden Seeds specializes in investing in female inventors and female founders of companies. So putting all those together has been the principal way that we've been able to fund ourselves over the years. Another question?
Okay. So I have a question on your IP strategy, given that the foundational patent has probably, 5 or 6 or 7 years to go, and your business is almost ramping up. Have you had other development in IP?
So IP is really interesting. For us, to the extent that it's possible, most companies, you actually have an interest in keeping things more trade secret than necessarily patent. Because as Kristala pointed out, patents only give you the right to sue. It actually won't stop someone. And you typically actually have to have evidence that they've encroached upon your patent. So if you consider, take our DSP process that we developed, that we've shared no patents against, once we publish that, if someone in China decided to build a factory using that technology, we'd have to know that they're actually using it, and there's no reason for us to know per se. So for us, all the developments, I said were, depending on how you measure it, 10x or 30-fold improvements. We've kept that all to ourselves. The only reason that we sort of would have to actually patent things rather than keeping them trade secret would actually be for investors, because investors sort of like to see that. But if you actually speak to large chemical companies, their best stuff, they don't patent. So it's really sort of a fascinating thing. So I had a conversation with, used to be the CTO of Praxair, and he started with, oh, what are your patents? And are you patenting more? And I said, but if it was yours, and when you were at Praxair, how did you think about these things? He was like, oh yeah, we usually try to keep things trade secret rather than patents.
There's a question online that's related to this question. What exactly is covered by your patents? Was the technology that was developed at MIT still related to all of your products?
Yeah, so the patent fundamentally cover, answer in the two ways. There are patent families. So there are patents where the claims are slightly different in different countries based on how the prosecution went in those countries. But the patent fundamentally covers a biological process for the production of glucuric acid. As Darcy was just saying, and this is true for chemical companies, but it's especially true, I shouldn't say especially, it's additionally true when you're talking about fermentation. that a lot of the process is where it's hard, and it's harder to find answers from that through the literature, right? So the patents don't cover the process. Trade secret covers the process. And the patents fundamentally just cover the demonstration that you can produce this product in a cellular system. Again, depending on where they are, it goes into multiple host organisms and multiple variants of the different enzymes that we use. And that is all based on work that was done at MIT.
So I would, along those lines, just in terms of patents, for some of the applications, I mentioned Professor Erica Ford at NC State. So we licensed the work that she did around glucaric. So that's a new set of patents that we have access to in terms of what we cover. The other just generic comment, within the field of fermentation, if you look at the patents and you look at the titer, so the concentration, which is one of the super important variables, the ones you see in the patents are, I said for ourselves, it's more order of magnitude off. That's true of most of the patents in this space. You'll see academic literature talking about things, but it's almost irrelevant in terms of actually commercializing, because it's all just about sort of proof of concept. But that, again, is where your advisors can come in and be very helpful, because your advisors will be able to say things like, yeah, well, the highest titer in the industry is probably around 150 grams per liter, or make generic comments like that, which you could scan the literature all day long, and you probably won't see that published, because the largest people in the space, Archer Daniels Midland, Cargill, they don't publish those types of results. You just have to have an insider who can give you insight into what the industry, wherever you are, thinks is typical or standard.
Which is better, okay. Hi, I'm Deirdre from the Technology Licensing Office. What would you have liked to have known in advance of you founding a company?
I'll let you start.
I'll go first. I think that the thing that surprised me most was, how do I phrase this? It comes back to the fundraising question that was asked initially in terms of what it is that people are looking for. And Darcy said it, and as an academic, this is very painful, which is that there are, and it's not universally true, but there are many investors who don't really seem to want to know the technology. There's a certain narrative that they have adopted, and they want to push you into that narrative. And so, that meant I would go on a lot of these discussions, and then after 5 minutes, people were talking about deal structures and cap tables, and I'm still not sure I understand what either of those are. So I think that was very different. And it actually reminded me, Darcy reminded me of this a few days ago. I had an experience very early on where I did some consulting from a company before I actually was involved in founding Calion, was hired as a consultant from a company, a very, very large company that had an investment arm that was looking at investing into a small company. And this was in the biofuels craze. And so I sort of listened to the pitch and asked these questions, and I went back to the investors and said, these are questions you need to know the answers to before you invest. And the response was sort of like, well, we don't want to make anybody angry by asking these questions. And my response was like, well, how are you going to give them these money without knowing the answers? So all of those kinds of things I saw coming back at our own efforts to raise funds, which is in the sense of how much are people trying to understand what it is that you're trying to do versus how much have they just decided, here's what I'm looking for, and you're either in that peg or you're not. And so another way I'll describe it is when we started kind of at the height or the depth, however you think about it, of the biofuels craze, right? where people were still investing in biofuels, where we were about 6 months away from the collapse. And people couldn't understand or wouldn't bother to understand how we weren't biofuels. They heard fermentation, you're not beer or wine, you must be biofuels, right? So I wish I had definitely I wish I had known that, because I think I would have been better psychologically and emotionally prepared for sort of the ups and downs of having these conversations where I'm an engineer, I'm an academic scientist, and I thought people would be really excited about the technology and want to understand. And again, it wasn't everyone. One of the most delightful meetings I was involved in was one where we had with an angel group, where someone walked in and had downloaded off of DSpace one of our papers. And I was like, this is gonna be fun, right? Because we could actually talk about science. But how quickly it becomes not about the science and technology and about all these other things. I had no idea about that at all, and certainly would have benefited from having a better headspace going in a lot of these conversations if I had understood that. You can give your answer.
No, so I'll just build on this in an interesting way. So when Kristala was coming back to MIT, so early noughts, right? So 2002, 2003, we're having conversations about, so what is she going to choose to do for her research? And I was like, so you're going to do biofuels? Because I'm reading the Wall Street Journal every day going, oh, there's like hundreds of millions, if not billions of dollars going into biofuels. So I was like, so you're doing biofuels? And just like that, immediately no. Now, if you know her, generally her responses are very thoughtful, very deliberate, especially her technical answers. I was like, okay, you said that really quickly, so you're very certain about it, so why isn't this going to ever work? And she goes, well, she said, I'm not doing it because it'll never work. I was like, what do you mean never? And then she goes, you started with never. And then she goes, well, it's at least a couple of decades. It's like, all right, VCs don't work on 2-decade timeframes. They work on 5-year timeframes. So what do you mean a couple of decades? How is this supposed to work? He's like, well, first off with biofuels, we don't know the science. It's like, we don't? Like, no. We do not understand the science behind how we're going to make biofuels competitive with traditional petroleum products. It's like, okay, so so how long does it take to figure out the science? Like, best case, 2 brilliant graduate students. So 5 years for the first one, 5 years for the second one. I was like, oh, that's 10 years. That's not 2 decades. Like, yeah, that's the science. Now you got to engineer it. And the engineering is often as hard as the science. So another 2 brilliant graduate students, so now we're 2 decades out. And that was roughly 2003.
It was 18 years ago.
And so we're still not there, but it's that sort of simple analysis that VCs can be absolutely brilliant and absolutely not brilliant, all at the same time. So that's one of the fascinating things that you'll see as you go through here, are the people you're like, they got it like that, and the people you're like, they have no idea what we're doing, and they weren't remotely interested in learning more about what we're actually doing. They just want to talk about markets. So, other questions?
Thank you, great talk. You're a company that essentially manufactures, and from the earlier, at least the story, it looked like for the very first couple of years you didn't have a lab, you didn't, have a physical asset where manufacturing was happening, or actual synthesis and validation was happening on a daily basis. So can you walk us through the stories of how you dealt with that, especially as you had to convince investors about the proof of concept, because I presume by the time you took the license for the company, you were not working on this in MIT anymore, right? So can you walk us through some of the stories around that?
So she did a little bit of working at MIT still, because there were some things that she could still do. There's some things that were no longer allowed. Basically, what we realized is that the infrastructure to do the scale-up experiments that needed to happen already existed. So there were places where you could rent that space in order to do it. So the initial investors invested because they said, she's brilliant. We don't know exactly what in the world she's doing because we've never heard of a story like this. Although actually Cherryston said, well, we did used to own a brewery. So we at least understand that aspect of it. They gave us the initial money so we could do the initial experiments, so we could do a lot of process optimization. And that facility already existed. And what we've essentially done since then is there are often very simple experiments you can do with a relatively small lab. So we've got limited lab space in Montana and San Diego, where we'll do the very simple basic experiment so we can know we need to run in that direction. And then we'll find an organization who's really good at running in that direction, and they'll do the proof of concept, the things that are beyond proof of concept to help us optimize those sorts of things. The other thing that we did that most companies didn't do was from day one, we actually modeled the entire manufacturing process. So we actually had a handle on what were the metrics that we needed to achieve for our titers and other lab scale things so that we could prove that it would actually work at the price points we were talking about.
Yes, I want to make a couple of comments because you mentioned, well, you know, at the time we started the company, we wouldn't have had manufacturing. At the time we started the company, we had me who still had a full-time, which I call my job at MIT, not my full-time job, my overtime job. So I had my overtime job at MIT. We had Darcy, who still was working for another company, and we had Neil Connors, who he mentioned, and that was it. And so I give TLO a lot of credit, and the MIT ecosystem a lot of credit for, I had that date there of April 3rd, 2009. That's the date where we started racking up debt to MIT without any guarantee that we'd ever be able to pay it back, right? So, you know, I think that's a uniqueness of this ecosystem, which is that there's the experience of TLO and the experience of this institution to be able to take calculated risks. Right? We've written checks to MIT since then, by the way. Right? So that's one. But then the other thing, so you made the comment, I wouldn't have still been working on in my lab. So I've had some version of some graduate student project that has involved glucaric acid continuously since 2004, or January of 2005. And they are not, and part of this is part of it is conflict of interest management, but part of it is actually what Darcy mentioned, which is that the things that are attractive for academic publishing are very different from the things that are required to get something to scale, right? So we've continued to use the pathway as a model to explore some other things. And so another way in which TLO has been very helpful is that early on, we sort of set up, every time we would transfer things to the company. So the company has a license, and with that license comes a responsibility for me as the inventor to provide materials. And then there are sort of different iterations and things. And it can be, as you can imagine, you know, quite a hassle to have to go through a new process every time. And so we did this a couple of times. And then Ben actually came up with the idea of just structuring a blanket MTA that, as long as it's still relevant to what the core license is, allows me, just by basically sending a note to Ben saying, okay, here are the strains we're going to send now to update, to provide updated samples that have sort of modest improvements. And so there's one of those in particular that doesn't show up in the patent, and I'm not sure anybody would sort of figure out, has now become, coming back to this trade secret, has become a key part of how the strains are constructed that Canon uses. And so even though we don't have any work directly related, every once in a while there's something that pops up where I'm like, hey, you know, we have this thing. It's not, it's actually in the public domain, so we're not giving Calion any early access to anything, but it's covered by the blanket MTA, and so that then allows us to more easily transfer those materials, and then they go into the whole pipeline that Darcy described in terms of having contract organizations to be able to run samples and do some of the scale up.
Since we're on the topic of working with the TLO, there's a question online. What was your process like licensing from the TLO? What was helpful and what was challenging?
All right, you're licensing.
So what was very helpful, I think, at least at that time, because organizations can change, was that TL had pretty much a standard way of licensing things, and they didn't try for every single inventor figure out exactly how much should the royalty rate be for this versus that, because all that would end up doing is consuming their time and our legal bills. And both of those things, you know, a top-notch firm in the Boston area is at least $1,000 an hour. So think about that. Just reading the patent, offering you no advice, but just reading that patent can be several $1,000s, right? So simplifying that process and sort of having a standard and saying, we're not gonna haggle every clause, you sort of accept the basics here or you don't. But those basics were very fair, they weren't onerous. When we would talk to folks about other institutions, there are a lot of nightmare stories of trying to get yourself through that process. And at that stage, you're like, law firms will defer the bills depending on which one you're talking to. And you realize, like, I have a greater debt for law than I have for experiments in my lab, right? And that's disconcerting as a startup. So MIT is terrific in that way. When you hear the other things, there aren't any substantial complaints. Yeah, sure, I'd love to have a lower royalty rate or whatever, but they really try to be extremely fair and transparent about the process.
There's another question online. This is for Darcy again. You mentioned that VMS was the best resource available at MIT for building a new venture. What specifically did VMS provide Callion in terms of introductions or early important feedback?
So some of the early important feedback was moving away from the technical to the markets. And I think a lot of undergrads here, I assume the graduate students already trained sort of day one to do that. But at that time, that wasn't a standard. The second thing was connecting us to our first investors. So we would not have gotten that connection to Cherrystone without VMS. And then it's just a general business perspective, right? So if you're able to get in, they'll give you a group of advisors and just the way that they see the world. Whether you agree with them or not, the important thing is to know you are gonna meet someone else who has that perspective, and you're gonna need to know how to manage it. So the fact that you can get different perspectives, especially perspectives beyond your lab group and your immediate environment where, this is amazing, this is awesome, start a company. But people will really ground you in terms of, well, if you're gonna do a company, here's what really needs to happen. Other questions?
We can change it a little bit, and this question is for Kristala. How did you go from industry back to MIT as a faculty member?
Painfully. So this is I was not expecting that question. So I'll answer this in a couple of ways. One is I'm an engineer by training. And I start by that because there's a cultural difference between engineering and science, where science is much more about discovery, engineering is much more about application. And that's a blanket description, and obviously there are engineers who do discovery-oriented work and scientists who do application-focused work. But I say that because it's not totally unheard of to have people who are academics in engineering who have had time in industry. Having said that, I knew it was not going to be trivial, and when I was still a graduate student, I actually did a little bit of, he talked about markets, I did some market research on the feasibility of going from grad school to a company and then back into academia without doing a traditional academic postdoc in between. And I got really wonderfully insightful advice from a professor named Harvey Blanch at Berkeley, who told me the two most important things were that if I were gonna do it, I had to go to a company that was known for research, right? So I couldn't go to a place where I would be like a process engineer. I had to be doing science, real science. And then the other thing he said was that you can't do it for longer than 5 years unless you want to be gone for at least 20 years. And it turns out there was a lot of insight in that second question that I didn't quite get. The first one was pretty obvious, which is that if you're going to become an academic, people want to know you've been on the research train the whole time and that you've never gotten off of that. The secondary side effect of that, though, was that because I went to a company that was very heavily focused on research, I also got to stay within my research community. So I got to go to conferences on a regular basis and still have interfaces and connections with people who are now my colleagues. That second part of it, in terms of where the sweet spot is really just about the culture of academia, and that in a company, it takes longer to build a reputation as a scientist that's known to the outside world. So the less than 5 years was a nod to the fact that you sort of look more like a traditional postdoc. The at least 20 years is best described as professor of the practice, right? So we have at MIT these things that are professors of the practice, which are people who have been out in the practice space, so in a company or in another environment, and are now coming back to teach about it. So I would say that's sort of the bigger picture in the sense of how the transition was made. The other aspect of that, though, is that it was, where I said painfully was the answer, is that if you work for a company, everything you do when you're at that company is proprietary. And as a matter of fact, if you look at the intellectual property form that you sign when you go to work for any company, it really says anything you think about belongs to them as well, right? It's both conception, ideas that you conceive, and ideas that are reduced to practice. So as a practical matter, that meant that whereas most other people who would have been in an academic postdoc, if you're a postdoc with Bob Langer, I can pick Bob because he's got 80 of them. If you're a postdoc with Bob Langer, when you go start your own lab, then you can take materials that you've been developing there and use that as the foundation for getting your lab up and running again. If you work at a company, when you leave with good wishes. And that's it, right? And I left with good wishes. I had a good working relationship. So that was the painful part of it, which meant that the research program I talked about was something I had to start from scratch. I had to obtain all the new strains and build everything up from scratch and wasn't really able to take anything with me. Having said that, I don't have any regrets at all about how I did it. For the purposes of this audience, what I will say is that I absolutely would not have had the framework to think about patenting and disclosures and the potential for commercialization had I stayed on the traditional academic track. My time at Merck was really influential and critical towards getting me thinking about the potential for outcomes from my academic research lab.
Thank you. You and Darcy touched a little bit on this about since you guys started Callion together, and then you were talking a little bit about biofuels and how your opinion has changed on it. But how has working in commercialization products changed your science, your research?
So he pulled back on me saying biofuels would never work. I didn't say never. I did say 20 years.
No, you did say never. And then I said, never? He said, okay, 20
Okay,
years.
right. So 20 years. So I think it's changed in a couple ways. So one, I mentioned, I'll sort of continue on the answer I was giving earlier, which was that the time that I spent at Merck also made me, I think, more purposeful about picking research projects in my academic lab that I think have potential for impact, right? And I have to be careful about how I say that, because I do fundamentally believe that in a well-managed academic research lab, everything has impact, even the things that don't work. Because at a minimum, what you're doing is training a group of really highly intelligent, motivated, dedicated individuals who will now exponentially go out into the world and make good things happen, right? So when I say at a minimum, that really is the maximum. It's why we do what we do. But it gives me this mindset in which I challenge my students all the time to be thinking about, why does this matter? What problem is it going to solve? How is it actually going to have utility? So as an example, for people who are in bio in the audience, you'll get it, others may not. There are fluorescent proteins that we use a lot to see how something is going. And the mantra is, we're never going to publish a paper where the only output is a fluorescent protein. We don't do it. We have published many papers where you use fluorescent proteins to show that something is working, and then you take that thing and apply it to a real system. But we're just never going to stop at the output just being fluorescent protein. And that's really, it is the combination of the time I have at Merck, but also this fact that I am, I live with this guy, right? So I know the kinds of challenges that Callion is going through, and I have a better sense of what are the real problems and what are the problems that are sort of toy problems. And a lot of the real problems, I can't see a clear path to try to solve them, right? So, there are still a lot of real problems where I don't necessarily see how we're going to solve them, but it does challenge me to be cognizant and to be purposeful about picking problems that I think are real legitimate problems, and not just sort of toy systems that may be interesting on the margins. I don't know if that answered the question, but it's at least what was on my mind.
Thank you. We'll take one last question from the audience. Okay, well, there's one question online, so I'll ask that if no one else has a burning question. Okay, the last question is, can you talk a little about the strategy of when you transition from lab to the company?
So I'll give the first part of that answer, and then I'll have Darcy pick it up. You know, the simple part is that the strategy was dictated by that ticking clock, right? That we had filed the provisional patent, and I didn't file the provisional patent with the idea of starting a company. But then the clock started ticking, and when the TLO email then came and said, do you want to do something with this? And maybe I'll go back and say, before we came to MIT, before I came to MIT as a faculty member and we moved here, Darcy has been very engaged in MIT since the time that he was an undergraduate. And so he had an idea that maybe I do something that could turn into a company well before I did. And so I kind of said it as a joke, was like, okay, well, if there's something that I come up with, you get to create the company and run with it, right? So when the email from TLO came up, I immediately just forwarded it to him and said, okay, what do you think? And I was a little bit surprised when he came back and said, I think you have something here. Maybe we should start a company, right? So, you know, I think if there is going to be an act two, I certainly know enough now to not have the patent timeline be driving it all. But that certainly was the first impetus, was the fact that we had to decide by April 4th of 2009, do we go forward with a PCT application or not?
And just to add a little bit of a nuance there, and Ben can correct me if it's still true. So one of the issues was MIT is fairly comfortable taking care of the US patent without a company behind you, but taking care of the international patents is what they don't want to do without there being a company behind it. So, but in our space, there are lots of significant players who do fermentation of products who are not in the US. And in fact, most of the folks who are really good at the industrial side of biotech are not in the US. And so that ended up being really compelling. And that is likely to be true for some of your other inventions, no matter what the space, right? You can imagine you come up with a new battery technology. Like, whether you like it or not, you're gonna get the cover of Nature for this breakthrough that's gonna save the world, and that clock starts immediately. And you can give up the rest of the world and just take the US, in which case the rest of the world's just gonna take the invention and run, or, you know, you have to think about your patent strategies. It's an unfortunate thing. Now, the nice thing is, once you're in a company, you don't have that same timeline pressure because you have no pressure to expose it to the world at large, right? It's the publicly speaking about your technology that's the trigger there.
Wonderful. Well, thank you both so much. Let's give a round of applause for Kristala and Darcy.